Cell structure and function. Every multicellular organism is composed of cells, which are the basic unit of life. The cell can be linked to a factory, and a factory has several mechanic machine which are linked to one another at a specific order, and each one make a particular component. The component of these machines are assembled to produce the desired product. Similarly, the cellular constitute, each with the specific function, have definite arrangement. They produce the component, in this case molecule, which are assembled to synthesize the required product, macromolecule. The function of organism as a whole is a result of combination of activity and interaction of the cell unit in the body. Hence, to understand the essential physiology of animal, we need to know physiological function of the cell. The cell, which is a fundamental unit of life. The living cell perform all the function of the life, such as intake of nutrient, metabolism, growth, reproduction. To perform this life activity, the cell has its various cellular constituent and organelles. Electron microscope have been available in 1940, and it paved the way for more specific knowledge of cell structure and the structure of organelle within the cell. It permit magnification of 10,000 times or more, and that is down to the molecular dimension. It has revealed the strict and orderly pattern of arrangement of macromolecule consisting the organelle of the cell. Hence, with the electron microscope, it is possible to observe the structural pattern of organism. The electron microscope and the ultracentrifuge have helped in merge of cytology and biochemistry and solve many physiological problems of the cell. The cell consists of a cytoplasm, which is an active fluid medium that helps us to carry out its life activity. Cytoplasm is a colloidal solution mostly containing water. About 30% of the total mass of the solution consists of various substances. Of these, 60% are protein and the remaining are carbohydrate, lipid and other organic substance and inorganic matter. The cytoplasm is enveloped by a membrane known as plasma membrane, and the plasma membrane is often termed as cytoplasmic membrane. Cytoplasmic matrix is a ground substance. 3 decades ago, only the cytoplasm and the nucleus were known to be enveloped by the membrane. So, with the advent of electron microscope, it was found that various organelle and inclusion floating in the cytoplasm are enveloped by membrane and separated from the cytoplasm. Commonly represented organelle as well as inclusion which are covered by membrane are the plasma membrane, rough ER and smooth ER. Golgi complex, lysosome, mitochondria, nuclear envelope, centriole, phagosome, pinocyte, vesicle, etc. Cellular membrane of the cell help regulating the passage of the substance through them, and such a passage may be passive diffusion or by active transport involving the aid of enzyme, which are located in the membrane. Another important function of the membrane is to provide a surface for harboring the enzyme. Nucleus is the most conspicuous structure in the cell. Usually, each cell has one nucleus, but cells such as liver cell and skeletal muscle cell contain more than one. The fluid matrix of the nucleus is known as nucleoplasm or karyoplasm. It is enveloped by a double layer nuclear membrane, and the karyoplasm has a densely stained particle called a nucleus. It is a large ingrowing cell and disappear during the cell division. Sometimes the nucleus may have more than one cell, and the karyoplasm is not generally different from the physical or chemical property of the cytoplasm. The most important content of karyoplasm is the chromatin, which is a combination of protein and deoxyribonucleic acid, that is the DNA. It is a granule in nature, but during cell division it is transformed to the long strand called chromosome. Fimbriae, in prokaryotic only. The structure, a hair-like structure on the surface of prokaryotic cell. The pila facilitate DNA transfer between bacteria, conjugation, and fimbriae aid in adhesion of the surface. These structures collectively maintain the cell's integrity, facilitating its function and allowing interaction with its environment. If we see the plasmid, in prokaryotic, the structure, it is a small circular DNA molecule. It separates from the chromosomal DNA. Its function is to carry gene for antibiotic resistance and other trait, and can be transferred between the bacteria. If we see the nucleoid prokaryotic, the structure irregularly shaped region containing circular DNA. Its function is to store genetic material in prokaryotic cell. If we see the flagella and cilia, the structure, it is made up of microtubule, flagella long and few in number, cilia is short in number, and the function of flagella is to propel cells, example sperm cell, and cilia move substance along the surface, example respiratory tract. If we see the cell wall in plant cell, fungi and bacteria, the structure, it is rigid layer outside the plasma membrane. It is composed of cellulose, plant and chitin, that is the fungi, and or peptidoglycan in bacteria. The function is to provide the structural support and protection, and it maintains the cell shape. If we see the centriosome or centriole, which are present in the animal cell, so the structure, the centrosome contain two centriole made up of microtubule, and their function is to organize microtubule during the cell division, and it forms the spindle fiber that separate the chromosomes. If we see the cytoskeleton, the structure is the network of protein, filament and tubule. It is composed of microtubule, microfilament and intermediate filament. So the microtubule, it provides the shape and guide organelle movement, and the microfilament support the cell shape and enable movement, and intermediate filament, it provide the mechanical support. The function is to maintain the cell shape and facilitate intercellular transport and cell division. If we see the chloroplast, that is present only in the plant cell, the structure, it is a double membrane structure which contains thylakoids that is stacked into the grana and stoma. The thylakoid have chlorophyll to the green pigment. The function is to conduct photosynthesis, converting sunlight into glucose and produce oxygen as a byproduct. If we see the vacuoles, the structure consists of a large membrane bounded sac, particularly prominent in the plant cell. The function is in the vacuole is to store water, nutrition and waste product. It maintains the turgor pressure in the plant and cell for the structural support. If we see the peroxisome, it is a small membrane bounded organelle. It contains oxidative enzyme. What is the function of peroxisome? To break down fatty acid and amino acid, and it detoxify harmful substance like hydrogen peroxide. If we see the lysosome, it is a membrane bound sac containing hydrolytic enzyme. Acid interior, pH that is 5, and the function is to digest macromolecule, older organelle and pathogen involved in autophagy, that is the self degradation. If we see the ribosome, it is made up of RNA and protein. It consists of two subunits, large and small, and the function is to synthesize protein by translating mRNA. Free ribosome produce intercellular protein. That is the endoplasmic reticular bound ribosome make protein for export or membrane. If we see the Golgi apparatus, the structure, it is a stack of flattened membrane bounded sac. So the Golgi apparatus, we know it has a cis receiving and trans exporting phase, and the function of this Golgi is to produce lysosome and secretory vesicle, and it modifies, sort and package protein and lipid. Now if we see the that is endoplasmic reticulum, it have two types, rough and smooth, and it synthesize lipid, detoxify drug and store calcium. The smooth endoplasmic reticulum consists of calcium, and the function of the endoplasmic reticulum, it acts as a manufacturing and transport system within the cell. If we see the mitochondria, it is known as the powerhouse of cell. It double membrane consists of double membrane, outer and inner membrane, with fold core cisternae, and the matrix contain enzyme, mitochondrial DNA and ribosome. The function of mitochondria is to generate ATP through cellular respiration, and it regulates the programmed cell death, that is the apoptosis. If we see the nucleus, it is surrounded by a double membrane nuclear envelope with pore. It contains nucleoplasm, chromatin, which is made up of DNA and protein, and the nucleolus. The function is to store genetic material and coordinate activity like growth, metabolism and reproduction. Nucleolus synthesize ribosome, that is rRNA, and assemble ribosome. If we see the cytoplasm, it is a gel-like substance containing cytosol, water, which contain this gel-like substance cytosol. What does it contain? It contain water, salt, organic molecule, and the cytoplasm, it is suspends organelles, and the function of this cytoplasm is the site for biochemical reaction. Here all the biochemical reactions occur. It provides the medium for intercellular transport. If we see the plasma membrane, it is a wide topic. Plasma membrane structure, it is composed of phospholipid bilayer with embedded protein, cholesterol and carbohydrate, and semi-permeable membrane. We know that the plasma membrane is a semi-permeable membrane, and the function is to protect the cell by acting as a barrier. It regulates the entry and exit of substance. Example, nutrient, waste, ion. It facilitates cell communication by receptor protein. Latest it is used in lipid rafting. These microdomain in the membrane are the key to signaling and pathogen entry. And membrane tension play a role in the process of like endocytosis, exocytosis and migration. Thanks for watching. Do like, share and subscribe.